The University of Dundee is one of the leading universities for scientific research and supports many different projects within the recently built Wellcome Trust building. Many research projects are very successful and many papers have been published from the discoveries made within the university. We have recently been interviewing Dr. Alan Prescott on one of his research papers investigating the cellular response of the metal binding properties of metformin. Anti-hyperglycemic guanide metformin is an extremely useful oral anti-diabetic drug used in the treatment of type 2 diabetes, particularly in overweight and obese patients, and with patients with a normal kidney function. When cells are treated with metformin, there is a reduction in the mitochondrial inhibiting complex 1, which then induces the activation of AMPK-sensitive signalling and suppression of the hepatic gluconeogenesis. These effects of metformin on gluconeogenesis are believed to be an important factor in metformin's anti-hyperglycemic properties. This paper was particularly interested in the metal binding properties of metformin as it acts as a ligand to a variety of transition metals with a valency of 2, the most stable of which being copper, an important regulator of cardiac health.
Hello there. We're here with Dr. Prescott to discuss his involvement in the cellular response to the metal binding properties of metformin. Can you explain the core science of the paper to us?
The main point of the paper really is that the drug metformin, which has been used for treating diabetics for a very long period of time, been known for a long time. The mechanism of action is not really fully understood. So the point of the paper was to try and find out what the mechanism of action of metformin is.
Sure.
AMP-activated protein kinase, or AMPK, exists as a H dimer made up of a catalytic subunit shown in orange, a beta subunit shown in blue, and a gamma subunit shown in green. Each of these subunits has a specified role in how stable and active AMPK is. When concentrations of AMP are increased and phosphorylation of AMPK on the threonine 172 activation loop, a conformational change in the gamma subunit expresses the active site at position 172 on the catalytic subunit. AMPK regulates many intracellular pathways like the uptake of glucose, beta oxidation of fatty acids, and biogenesis of glucose transporter 4 and also of the mitochondria. During rest or exercise, AMPK reacts to changes in the AMP to ATP ratio. When the muscles are stimulated, AMP levels increase while ATP levels decrease. This causes a conformational change and AMPK becomes an excellent substrate for activation. AMPK activity increases during exercise due to the muscle cell being put under stress due to the high demand for ATP in the cell. Inhibition of anabolic energy pathways by AMPK increases the energy in the cell. While investigating metformin's metal binding properties, it was found that its ability to bind with the transmission metal copper is somehow crucial to the effects on the AMPK pathway. This was proved by using the metal sequestration molecule triene. Triene's actions as a metal sequestration molecule mean that it readily binds to copper in the mitochondria, reducing the free copper in the cell. H411E cells were pre-treated with triene and were shown to produce punctuate fluorescent in its presence, providing the lack of free copper in the cell. However, this reduction in available copper means there was less for metformin to bind to. Thus, it was found that metformin's actions were opposed and the AMPK pathway was inhibited, proving that copper is necessary for metformin's effects. Metformin therefore needs to be able to compete for copper in the cell, as it has less copper binding sites displayed on its surface than that of triene. It does this by means of pi electron delocalization. This was proven by use of copper probe again, by which metformin produced punctuate fluorescence just as readily as triene, but molecules lacking in pi electron delocalization had no effect. The mitochondrial respiratory chain complex is the primary target of metformin. Metformin's intracellular transport is mediated by organic cation transporters, as it can only marginally cross the plasma membrane by passive diffusion. Once inside the systolic compartment, mitochondria then constitute the primary target of metformin. It has been shown that the bigonide drug inhibits the mitochondrial respiratory chain, specifically at the complex 1 level, without actually affecting any other steps of the mitochondrial machinery. a decrease in NADH results, and ultimately a reduction in proton-driven ATP synthesis. This decrease in ATP results in transient inhibition of energy-consuming gluconeogenic pathway in hepatic cells. Metformin targets the expression of glucose 6-phosphatase, and inhibition of this enzyme affects the production of fructose 1,6-biphosphate levels, and hence reduces glucose production. With analogues of this drug, such as PDI, opposite effects have been observed, showing an increase in hepatic glucose production and glucose 6-phosphatase expression. This highlights the importance of the structure of metformin and the anti-hyperglycemic action of metformin. Metformin targets complex 1 in the mitochondria and reduces ATP levels, which has an effect on different pathways. As a cellular energy sensor responding to low ATP levels, the AMPK phosphorylated activation positively regulates signaling pathways that replenish cellular ATP supplies, including fatty acid oxidation. AMPK negatively regulates ATP-consuming biosynthetic processes, including gluconeogenesis, as shown before, and lipid and protein synthesis. AMPK activation reduces hepatic glucose production by inhibiting lipogenesis and indirectly increasing insulin signaling. Overall, metformin acts to inhibit gluconeogenesis.
Metformin is obviously prescribed to a lot of people with diabetes, and it certainly would be a worry to me as a member of the public that people aren't absolutely sure of the mechanism of action. That's one thing that we would really like to understand, and that may give us an advantage, because once you understand the mechanism of action of the particular drug, it then becomes much easier to design new drugs which may be related in structure to this compound that may actually be more effective, have less side effects and so on. So it's a way into future treatments. So it's not really an end point, it's a starting point really for the project.
Are there any sort of further studies potentially in this to come from this research?
Yeah, sure. Since this paper was published, we've managed to get a grant from the MRC funding agency to do further research into metformin and related compounds, with a view to trying to understand the real mechanism of action of metformin, and again to then use related compounds to see if they have similar or different actions.
This study found that the effects of metformin and its analogues were dependent on their affinity with copper. This research opens doors for potential investigations to be carried out on other compounds that could form pseudo-aromatic planar complexes with copper, or more generally, the role that the metal ions have in chemical bonding and drug structures.